My name is Kate Dickerson, and I am the founder and the director of the Maine Science Festival. And just a quick overview for me before I turn it over to our speaker. First of all, we're running these sessions, these forums once a week for the entire month of March. I'll have more information at the end about what you can expect coming up. But for us, March is Science Month, so we claim it. And we're taking it over, and in this case, in our pandemic times, that means a forum. So I am delighted that we're able to do this. We're able to do this because of the sponsorship provided by the Bioscience Association of Maine and the Jackson Laboratory, as well as the partnership of Maine Public, who's been helping spread the word. COVID-19 is going to be the focus of every session in one form or another for the next 5 weeks. And that is because it's a science story of our times. There's just no way around it. It's going to impact us for at least a generation, if not more. And no matter what you think about it, the science that we have learned and used has been just extraordinary. And so without any further ado on that piece of it, because Melissa, I believe, is going to cover just a little bit of that, I'm thrilled to introduce Melissa Maginnis, who's a researcher and associate professor at the University of Maine. She also has another title that I'm not going to get correct. I think it's the Associate Director of the Center for Undergraduate Research. Did I get that? Yes. All right. And then also, she's the scientific lead for the COVID-19 Advisory Board for the UMaine system. So Melissa is going to provide an overview of COVID-19 as a virus itself, which I think is going to be wildly interesting. Hopefully we'll all have a better appreciation of how things happen the way they have. As Melissa goes on, please feel free to put questions in the Q&A box. Once we're done, Melissa's going to present for about 30 minutes at the most, and then we're going to open it up for questions. So please get your questions ready, and Melissa will do everything she can to answer them. And if she can't, neither one of us has a problem saying we don't know. So we'll be happy to let you know that we don't know. So without any further ado, I'm handing it off to Melissa.
Awesome. Thank you so much, Kate. And I'd like to thank Kate and the Maine Science Festival for the invitation to speak today. It's really such a pleasure to be here, and it's absolutely the virus and the science of our times. And I'm so happy to see that the Maine Science Festival is continuing to go on, because this is a time where we really need to understand science, and we need to celebrate all of the wonderful discoveries that have been made thus far in COVID-19 area of research, and also learn how to communicate science effectively. So I'm really delighted to be here with you today and tell you a little bit about what I know about viruses. So I'm going to share my screen with you. Hopefully you can all see it. Just a second there. All good? Okay, so today I'm going to provide an overview of COVID-19 and the virus that causes COVID-19, SARS-CoV-2. And I have been studying how viruses infect cells and cause disease for about 20 years now. And I find this process to be absolutely fascinating. And one of the reasons that I got into this field is because I wanted to help relieve human disease and suffering. We know that viruses can cause significant human disease, and we've seen how COVID-19 can change every aspect of our lives. And one of the reasons that I was inspired to go into this field is because my grandfather was a polio survivor. And when he was 21, he was paralyzed from the neck down when he was infected with poliovirus. And we don't really hear about poliovirus very much anymore because we have a very effective vaccine that can prevent poliovirus disease. But growing up, I was able to see the significant impact that that virus had on his entire world and how it really impacted the way in which he saw medicine and the way in which he was able to walk, and his legs were really weakened by that disease. And so when I saw how the virus could cause such havoc on someone's body, it really made me interested in studying viruses. So we've all heard about COVID-19, and the COVID-19 pandemic has really changed the way in which we are interacting in the world over this past year. So COVID-19 started off being called novel coronavirus when it first emerged in 2019, December of 2019 in Wuhan, China. And at that time, we knew very little about the novel coronavirus. And over the past year, we have seen remarkable discoveries surrounding this virus. We understand so much more about the novel coronavirus, now called SARS-CoV-2. We know how the virus is spread. We have developed diagnostics to test for this virus. There have been treatments developed, and we have 3 vaccines with emergency use authorization that have been emergency use authorized by the FDA for prevention of COVID-19 disease. So this is really remarkable. And I think it's interesting to just think about where we were about a year ago. And on March 11th of last year is when the World Health Organization declared COVID-19 a pandemic. So around this time last year, we were all hearing that this is a virus that was causing pandemic proportions. Schools were starting to go remote. Some of the schools never went back in person. In the state of Maine, we've been pretty fortunate that we've been able to be in person, at least to some extent. It's changed how we've interacted with one another, our social interactions. It's changed our education. It's had a significant impact on the economy. And so we can see that all of this has been caused by a virus, a single virus. So if we look at where we were about a year ago, we were facing things like PPE shortages. We didn't really have testing available. So even if someone had COVID-19, we didn't have good testing available to accurately determine whether or not they were infected. There was a significant burden on the healthcare system. And we had so many questions that weren't answered. Some of these questions were things like, how does COVID-19 spread? Everyone thought that there was a significant amount of fomite spread that was going on. And people were holding all of their mail for a couple of days and washing down their groceries because we didn't know how COVID-19 was spreading. We didn't know how many people were going to contract COVID-19. And then how are we going to test people for the virus? How are we going to treat them? What's the mortality rate? What are the long-term effects of this illness? Will people develop immunity? We had no idea that we had a vaccine on the horizon. We knew that there were many companies working on a vaccine, but usually it takes 5 at the best, sometimes 10, 20 years to develop a vaccine. And we have diseases like HIV for which we still don't have a vaccine. So I think we were all hopeful, but none of us really dreamed that we would have vaccines available within 1 year. So we were all called to flatten the curve. And we were able to do this pretty effectively, in which we were trying to reduce this peak in the outbreak and try to reduce the burden on the healthcare system. And if we think about how this all started, in 2019, there was the first person that developed symptoms of the Wuhan coronavirus in Wuhan, China. And this started in the beginning of December. And then at the end of December, China alerted the World Health Organization about these pneumonia cases that were popping up in China. And it was linked back to a seafood market in Wuhan, China, and that was shut down. And then January 11th, we had the first death that was recorded. In that period of time, between the first patient developing symptoms and the first death, it was about a month. And during that time, it was identified as a new virus called COVID-19. Well, the disease is called COVID-19. The virus is called SARS-CoV-2. And we started to see cases in the United States, right around January of 2020. And from here, the death toll climbed. And this is just showing you the beginning, the snapshot of the COVID-19 pandemic. But let's look at the current situation. So worldwide, there have been over 114 million cases, over 2 million deaths that have been caused by COVID-19. In the United States, there have been over 26 million cases, and we have just reached a critical milestone of 500,000 deaths in the United States. In the state of Maine, there have been over 44,000 cases and 703 deaths. And what is really remarkable to me is every time I speak about COVID-19, SARS-CoV-2, I have to update these slides, and the numbers just continue to grow. So although we have learned so much about COVID-19, this virus is still continuing to spread. And so I want to take some time and talk about what is a virus and talk about the infectious cycle of SARS-CoV-2, and then talk a little bit about the science and the transmission of the virus and what makes it unique and what gives it pandemic potential. So first, starting off, viruses are tiny yet powerful. And if we look at this comparison chart, we can see the size of viruses. Some examples are shown here, like a bacteriophage that infects bacteria, smallpox virus, and then some of the teeny viruses like poliovirus, in comparison to an E. coli bacterium and in comparison to a red blood cell. Okay, so this is the red blood cell compared to the E. coli. And if we zoom in on that, that's where the viruses are. And we keep zooming in. And we can see how small they are in comparison to a bacterium or to a red blood cell. And I think that this is a pretty cool little tool that we can use to look at the size and scale of viruses. And I'm just going to pop this over here on my screen. And this is something that you can play with. So when we talk about the size of viruses, we usually refer to them in nanometers. And nanometers are 10 to the minus 9th meters. We're all familiar with a meter and a centimeter and probably even a millimeter. But a nanometer is 10 to the minus 9 meters. Okay, so if we start off looking at coffee bean, that is about 12 by 8 millimeters. And then we zoom in, we see a little sesame seed there. It's about 3 millimeters. And we keep zooming by the grain of salt and an amoeba. And we even get into a skin cell. It's 30 micrometers, 10 to the minus 6 meters. And the red blood cell, and we keep going smaller and smaller. Oh, we reach the E. coli bacterium. And the E. coli bacterium is about 3 micrometers or 3,000 nanometers. And then we keep going, and then we start to see viruses. And the viruses that we see are things like measles virus, influenza, HIV. And SARS-CoV-2 is about the size of HIV and influenza virus. It's somewhere around 100 nanometers in diameter. So there are viruses that are even smaller than this, like rhinovirus, which causes the common cold, which is only 30 nanometers in diameter. But you can see just how small viruses are. And they are the smallest of all microbes. And they're said to be so small that you could fit 500 million of those rhinoviruses that cause the common cold on the head of a pin. So of course they're microscopic. And so we don't know when viruses are surrounding us. We don't know when they're spreading because we can't see them. So let's talk a little bit about the basic virus principles. What is a virus anyway? So here are some images of viruses, and I think that they are just striking. I think that science is a bit of an art, and to me, this is an art. Viruses are made up of a nucleic acid genome that can be either DNA or RNA. Just like our genetic material is made up of DNA, and the DNA encodes for genes that ultimately make up proteins, viruses are made up of either a DNA or an RNA genome. That genome is surrounded in a protective protein coat known as a capsid, and that's illustrated down here. And we see that protein capsid can be further surrounded by a lipid envelope, and that would be an enveloped virus. Viruses that don't have an envelope are called non-enveloped, and sometimes they're called naked viruses. And virions have to be metastable particles. And what that means is that they have to be stable in the environment because they have to be able to withstand things like changes in temperature and pH, and they have to be able to stay together to infect the host cell. But then once they infect the host cell, they have to be able to come apart and deliver their genetic material into the host cell. The really interesting thing about viruses is that they can only multiply inside a host cell. So they need a host cell in order to replicate. And that is different in comparison to a bacteria that can replicate outside because it can replicate on a surface, it can replicate on your skin, but a virus actually has to infect the cell, and therefore it's called an obligate intracellular parasite. If we look at the overall structure of SARS-CoV-2, we can see some of these unique features of the virus. So this is an enveloped virus. You can see the envelope is shown here in red. And then there's a spike glycoprotein, and that's sticking up on the surface of the virus in these little yellow projections. And spike is a really important protein because this is the viral attachment protein. It allows the virus to attach to our cells. And it's also a very important component because it's what the immune system can recognize to make antibodies to. So those spike glycoproteins, you can see them sticking out from the surface, and they look like little finger-like projections. And they allow for the attachment of the virus to the host cell. And then once the virus is able to attach to the cell, it can then invade the cell and deliver this genetic material into the host cell, which can then be replicated. And at the beginning of the pandemic, I made a model of this virus with my then 9-year-old daughter. And we did this using a little clementine and some frilly toothpicks. And so if you think about the clementine being the lipid envelope of the virus, and then the frilly toothpicks are the spike-like proteins. And these little projections are what allow for binding of the virus to the host cells. And we were doing a little lesson on how a virus could cause pneumonia by infecting cells in the lung. And we talked about how they bind to specific receptors. So we're going to talk about that in just a sec. So the coronavirus virions are called coronavirus virions because when they're analyzed by electron microscopy, they give off this little corona or crown-like appearance. And that's why they're called coronaviruses. And we can analyze the structure of coronaviruses using things like electron microscopy techniques. And using these techniques, we can appreciate the overall structure of the virus. We can see that the virus has a size between 60 and 140 nanometers. And the reason why there's a range is because these viruses are actually pleomorphic. And so they have some slightly different sizes and shapes. But overall, the mean size is about 67 nanometers in diameter. And that compares to SARS-CoV-1, which SARS-CoV-1 is the most closely related virus to SARS-CoV-2, and it's slightly larger. So when SARS-CoV-2 infects a host cell, it has to take over the host cell and turn it into a viral replication factory. And all viruses have to do this. As I said, viruses have to replicate inside a host cell. And I think that this is one of the coolest aspects of virology. So a virus has to first find the host cell, it has to attach to the cell surface, and then it has to cross over the plasma membrane. It can do that by using the machinery of the cell or by directly fusing with the plasma membrane. Then it has to traffic to the correct compartment in the cell. I talked about viruses being metastable, and it has to find the right compartment before it comes apart. Then when it comes apart, it can deliver the genetic material into the proper compartment within the cell. And that genetic material then serves as the instructions for the virus. It's essentially a parts list for all the viral proteins that need to be made. And it also serves as the template to replicate the genome. And when the genome is replicated, we can think about that as just like a piece of paper on a copy machine, and it's just making copy after copy. So it's making lots of copies of the viral genome. It's also using those set of instructions, that parts list, to make new viral proteins. And then those newly replicated genomes get encapsulated in those newly formed viral proteins to make new virions that get released from the cell, and those can go on to infect new cells and new hosts. The way in which SARS-CoV-2 is able to achieve this is utilizing that spike glycoprotein that sticks up from the surface of the virion. And spike adheres to cellular receptors on the cell surface. So these are proteins. They are not there just for the virus to bind to them and to mediate virus internalization into the cell. These are proteins that serve other functions that the virus has hijacked for its own use. The virus binds to these receptors called ACE2 and TMPRSS2, uses these receptors to attach and enter into cells. Once it has entered into cells, the virus starts to uncoat, and then it releases its genetic material. The genetic material for SARS-CoV-2 is a single-stranded RNA genome. So that's its little set of instructions, and that serves as the instructions to make these new viral proteins and also to copy the viral genome. Within the cell, this takes place on these structures that are formed from the endoplasmic reticulum. It forms these membrane-bound vesicles that serve as the site for viral replication. Once the viral genome has been replicated and those proteins have been produced, then the newly formed viral genomes get encapsulated and they can be released from the cell. An interesting thing about SARS-CoV-2 when compared to SARS-CoV-1, which was the original SARS coronavirus, is that they both use the ACE2 receptor and they both also use TMPRSS2. And of course, this is outdated at this time, but you can see some other interesting features. Both of these viruses emerged in the winter months, but SARS-CoV-2 did not cause a significant outbreak. It caused about 8,000 cases and around 774 deaths. And SARS-CoV-2, as we know, has caused much more significant pandemic proportions. So what leads to these emerging and re-emerging viruses? So in addition to the SARS-CoV-2 outbreak, in the past couple of decades, we have seen other emerging and re-emerging pathogens. We are probably all familiar with the swine flu outbreak in 2009, and this caused about 60 million cases and over 12,000 deaths. In the Ebola outbreak, there were about 30,000 cases and 11,000 deaths. Zika virus outbreak, which causes microcephaly in infants when pregnant mothers are infected with Zika virus. This occurred in 2015, resulting in 30,000 cases. And then the SARS coronavirus outbreak, which occurred in 2002, it caused 8,000 cases. And then MERS coronavirus outbreak in 2012 caused about 2,500 cases. And then we all know what has happened with SARS-CoV-2. So these viruses have resulted in really different outcomes, even though they share many commonalities. So just looking at these various SARS coronavirus outbreaks over time, as I said, the SARS coronavirus outbreak was 8,000 cases and MERS coronavirus outbreak was about 2,000. And then the virus kind of fizzled out. But we've seen around 115 million cases for COVID-19. And in addition to these coronaviruses that have emerged, there are other human coronaviruses that cause things like the common cold. And it's thought that these viruses may more commonly circulate in children, and children may have some immunity that provides some cross protection to SARS-CoV-2. Interestingly, with the SARS coronavirus outbreak, there was a physician who treated patients in China for a pneumonia outbreak. And then he traveled to Hong Kong and he died in the hotel the next day. And through his travel, he transmitted it to 10 other people. And then those people flew to places like Singapore and Vietnam, Canada, United States. And SARS ultimately spread to 17 countries. Similarly, the MERS coronavirus resulted in 2,500 cases in about 21 countries. But we know that there's been much greater widespread cases of COVID-19. So let's think about some of the commonalities and some of the differences between these viruses. So the low pathogenic coronaviruses are those that cause things like the common cold. And these viruses can circulate in either bats or things like mice. The highly pathogenic coronaviruses also have bats as a primary host reservoir. The SARS-CoV-1 is thought that it actually was transmitted from bats into a civet cat before infecting humans. For MERS coronavirus, it's thought that it passed into camels and then infected humans. With SARS-CoV-2, it's not totally understood yet, but it's thought that it was spread from bats into pangolins. And the reason why is because we see some unique features in SARS-CoV-2 that are not present in the bat coronaviruses. And it turns out that bats actually have lots of coronaviruses. So just like we have a microbiome, we also have a virome, and bats have their own virome. And 35% of the bat virome is coronaviruses. And people that have done surveillance, researchers have done surveillance on bat coronaviruses, and what they have found is that there are over 400 novel strains of coronaviruses that could potentially lead to a spillover event. And these bat coronaviruses can directly infect human cells. And the reason why is because the bat coronaviruses use that same receptor, ACE2, that the human coronaviruses utilize. And when there's a comparison in the amino acid sequence of SARS-CoV-2, and we compare the virus that was isolated from bats to the virus that was isolated from pangolins and to that with humans, we see that there are some unique features. And if you have never looked at an alignment of amino acid sequence, it is totally okay. You do not have to be a rocket scientist to understand this. And what I tell my students all the time is to look for patterns. You can identify patterns, even if you've never looked at a sequence alignment. So if we look at the human SARS-CoV-2 and compare it to the bat coronavirus and the pangolin coronavirus, you can see that some of the common features are highlighted with little boxes. And there are some that are common to the human and the pangolin coronavirus that are not seen in the bat coronavirus. So although the bat coronavirus has 96% sequence identity to the human coronavirus, there are these unique features that are only seen in the pangolin coronavirus. So this suggests that there may have been some zoonotic transmission from the bats to the pangolins and then ultimately to the humans. Now, how does this happen? Well, the way that this happens is because viruses are basically just self-replicating strings of letters. And the SARS coronavirus genome is made up of 30,000 letters that make up its RNA. And they have these imperfect internal spell checkers. And when they're making thousands of copies in a cell, they're bound to make mistakes. And coronaviruses actually make fewer mistakes than other viruses, but they evolve at a rate where they are generating about 1 to 2 mutations every month. So as the virus continues to spread, and as people continue to harbor the virus is going to continue to accumulate mutations. And then that virus is spread to another person and it continues to accumulate mutations. So you're probably hearing about variants in the news, and there are lots of variants. And these are mostly called variants of concern when they are shown to have some kind of increased transmissibility or increased pathogenicity. But you might be hearing about the New York variant or the California variant or Ohio, but those that are of major concern to us at this point are the UK variant, the South African variant, and the Brazil variant. And these are the other names that they go by. I'll refer to them as the UK, South Africa, and Brazil variant. So when you look at this coronavirus genome, and the genome here is coded in color, and it's showing you the various proteins that are encoded by those instructions in the genome. And if we take a look, for instance, at the UK variant, you can see these lines are showing you where the mutations have arisen in the coronavirus genome of the UK variant. And the majority of these mutations are clustered in that spike protein. And remember, that's a really important protein because that mediates attachment to the host cell. So in fact, all 3 of these variants of concern have mutations in spike. And it's a particular mutation at this asparagine residue 501. And that's just the location in the amino acid sequence. When this residue is mutated, it actually allows the spike protein to have a higher affinity for the ACE2 receptor, so it can bind to cells better. And this is thought to lead to the increased transmissibility that has been seen with these variants. And the reason why this is a concern is because in the United Kingdom, that UK variant became the dominant strain in the UK. And it's thought that if these variants are circulating in the United States, which they now are, that they could then become the dominant strain that's also spreading in our country. Interestingly, these variants have all arisen through what's called convergent evolution. So they have arisen independently of one another. And it's thought that there's some kind of selective pressure that's being put on the virus that's allowing the virus to have this adaptation that allows it to bind tighter to the ACE2 receptor and therefore become more transmissible. In addition to this mutation, these variants have other unique mutations. And so far, they have not been shown to be more pathogenic, but they are thought to be more highly transmissible. There could possibly also be some immune escape in the South African and the Brazil variant, meaning that the vaccines that have currently been developed may not be quite as effective against those variants. So how is SARS-CoV-2 transmitted? I think that we probably all know this by now, but when someone is infected with SARS-CoV-2 and they can release the virus through droplets when they are breathing, talking, coughing, sneezing, these droplets are released into the air and then they can land in someone else's nose, mouth, or lungs. They can fall onto surfaces or objects, and then if we touch those objects, we can then transfer them into our own nose and mouth. So of course, there's been lots of scientific evidence that has suggested how the virus is transmitted through droplets in the air. And there are, of course, great ways in which we can prevent the spread of virus by wearing face coverings, by physically distancing, by having strong hand hygiene efforts, and also by sanitizing surfaces. When someone becomes infected with SARS-CoV-2, they have a significant range of symptoms. And initially it was thought that these were only respiratory symptoms, that the virus was causing pneumonia, but actually it goes far beyond respiratory symptoms. People have, in addition to cough and shortness of breath, they have fever, they also have diarrhea. That was a hint that the virus is also infecting the gastrointestinal tract. Of course, they have things like chills and muscle pain, sore throat. And one of the really unique symptoms is this new loss of taste or smell. And that is something that's really unique to SARS-CoV-2. But now, as the fatality rate has dropped since we have increased the availability of testing, and we do have some treatments, and now we have vaccines, what actually is the major concern is the long-term health impacts. So there have been neurological symptoms that have been reported, continued shortness of breath over long periods of time, weakened heart, impaired mobility, and things like liver damage and lung scarring. And of course, we know there's increased risk for severe disease in people that are elderly and also those with underlying medical conditions like obesity and diabetes. And one of the things that makes SARS-CoV-2 so transmissible and so difficult to contain is that at least 20 to 50% of the COVID-19 cases are without symptoms. So some people may have one of those symptoms, some people may have a collection, and some people it may be so mild that they don't even know. And therefore, they can be spreading the virus without knowing it. And during that time, the virus can be incubating, and the person can be shedding the virus, transmitting it to others for up to 14 days. And at some point, they may become symptomatic, and in other cases, they may never show any symptoms. And then disease progression is very variable. Sometimes there are complications and sometimes people recover. And another thing that has been really challenging about understanding COVID-19 transmission is that it's difficult to model because of the way in which this virus is dispersed in disease clusters. So there are super spreader events that are thought to account for possibly 10 to 20% of cases, leading to 80% of the virus spread. And we also know that the risk is 20 times higher indoors. So there is a basic reproduction number, or an R0 value, which is a mathematical calculation that you can use to determine how many people will become infected if one person is infected with the disease, like COVID-19. And the reproduction number for COVID-19 is estimated to be about 2.5 in comparison to influenza, which is about 1.5. But the reason why this becomes so much more complicated is because of these super spreader events that can lead to possibly 80% of the spread. So the best way in which we can prevent the spread of COVID-19 is by using strong public health measures. The reason why we have to do widespread testing is because we can have 20 to 50% of people that are asymptomatic and spreading the virus and not even knowing it. Then when those individuals test positive, they need to be isolated, they need to stay home, and they shouldn't return to work or school until they're healthy enough to do so. We also have to do contact tracing on anyone that may have been in close contact with someone who is positive. And the reason why is because we want to make sure that those people are quarantined so that they're not spreading it, because that 14-day incubation period can lead to a lot of transmission if they're interacting with other individuals, and that can lead to significantly increased spread. But none of the public health measures that we have is efficient on its own. And I think that this is a really effective model to think about transmission and prevention of SARS-CoV-2, is this Swiss cheese model. So each slice of Swiss cheese has holes in it, just like each measure of prevention has holes. And so unless we put all of those slices together and we get a block of Swiss cheese, that's the only way in which we can really have a successful prevention of SARS-CoV-2. So we need physical distancing with good ventilation, with hand hygiene, face coverings, testing, contact tracing, and surface cleaning. And so we know that we're supposed to keep at least 6 feet distance, and that's based on the science. It's based on the transmission of the droplets that can be released from someone who is infected with SARS-CoV-2. So at least 6 feet distance. We also need to wash our hands. And the reason why we wash our hands with soap and water, or sanitizer if we don't have soap and water, is because it actually breaks up that lipid membrane that surrounds the virus. And once we can break up the lipid membrane, the virus is no longer infectious. And we also have to avoid the 3 C's: crowded places, close contact settings, and confined and enclosed spaces. And that's because we increase our risk as we increase the exposure time and the concentration of the virus. But throughout everything that we've seen in this past year, what we've really learned is that science is remarkable. It's been really amazing to see that in the matter of a year, we have identified a novel coronavirus, and there's been the development of 3 vaccines with emergency use authorization from the FDA. And so now we're moving into a time where people have access to the vaccine and will have more widespread prevention of COVID-19. And ultimately, we may move into a place that we can start to resume some level of normalcy. Some of this amazing research has been going on the campus of the University of Maine, done by my colleagues, Rob Wheeler, who has developed wastewater testing at the University of Maine to detect SARS-CoV-2 in wastewater, because as I said, it infects the gastrointestinal tract. Caitlin Howell, who is developing a bio-inspired membrane to understand how the virus is spread in high traffic environments. And also colleagues in healthcare, including Debbie Saber, to understand how the virus is transmitted in the healthcare system. So during this time, when there's information coming at us from every direction, how can we understand where to find answers to these critical questions? So we can look in the science literature, we can watch the news, we can listen to public health authorities, social media, and professional networks. These are all really good sources of information, but what's central to all of this is critical thinking. We have to be able to think through this information and to understand what is true and accurate information and what is misinformation. And using our critical thinking skills is really the only way in which we can navigate during this information overload. Some trustworthy sources that I want to leave you with is to look for the Center for Disease Control and Prevention, the World Health Organization, the Maine CDC, and our very own Dr. Shah. Look for information from the FDA And one of my favorite sources throughout the pandemic has been this dashboard that was developed by Johns Hopkins University that provides really up-to-date information. And if you seek trustworthy information sources, then you can know that the information that you're getting is accurate and up-to-date. So I'll stop there for questions, and I've included some resources here for the UMaine Science Advisory Board and my lab at the University of Maine, and you can feel free to contact me also if you have questions later.
Melissa, that was great. Thank you. We do have a few questions.
Mm-hmm.
Can you hear me?
Yeah.
All right. So one question was, how do the spike proteins on the virus help it infect other cells? And with that question is, can viruses exist without spike proteins?
Viruses cannot exist without spike proteins. So the spike protein that's sticking out from the virus is going to attach to the ACE2 receptor. And when it attaches to the ACE2 receptor and to the other co-receptor, that's going to trigger the cell to internalize the virus. The virus has to be internalized into the cell to ultimately uncoat and release its genetic material. So that attachment step is the first critical step in the virus infectious cycle. So if it's not able to do that, it's not going to be infectious. And that's a really interesting question, because if we have antibodies to that spike protein, that's how we're able to prevent infection. And the vaccines that are currently in use actually are the instructions to the spike protein. And when we produce the spike protein in our cells, then we have antibodies that are formed to the spike protein, and it prevents the spike protein from binding to the ACE2 receptor, so we can't get infection.
Okay, and then another question. Do vaccines play a role in creating more virulent viruses and strains, similar to how repeated vaccine use can create vaccine-resistant bacteria?
That is a really good question. So could the vaccines ultimately lead to the development of new viral strains that arise because they're generating mutations? And that is always a possibility. One of the ideas is that perhaps people that have long-term infection with COVID-19, these are so-called long haulers, that in those individuals that don't have a robust immune response, that perhaps we have mutations that arise in the virus that's replicating in those individuals, and that could give rise to variants. But in that setting, it's thought that it's due to a lack of a robust immune response. With the vaccines, the idea is that the vaccine would neutralize the virus before it's ever able to infect us. And so therefore, we shouldn't really have infection, and therefore we shouldn't have replication. So it should not lead to the development of these variants, but there is always a possibility if someone had an immune response that wasn't quite as robust. But I don't think that we would see widespread evidence of that.
Okay. Is it possible for the COVID-19 tests to improve to the point where they don't return false positives or false negatives? And a continuation of that, what is the difficulty with detecting the virus in human bodies, which I assume is one of the issues with those tests that we have right now?
Mm-hmm. Yeah, we've seen significant improvements with the diagnostics of COVID-19. So there are nasal swab samples, there's saliva samples, there are lots of ways in which we can determine whether or not someone is infected with COVID-19. And then generally, the gold standard in COVID-19 testing is an RT-PCR test. And that test is very sensitive. And so even someone who is asymptomatic, they would test positive. Now, of course, there always could be a false positive, but with an RT-PCR, there's a very low chance of having a false positive. And one of the things that they're doing with some of the saliva testing, for instance, is they're taking anything that's a presumed positive and then retesting it. And then they're essentially getting their false positive rate down to nearly zero. So if they are retested, you can have a false positive rate that approaches zero. So it sort of depends on the standard protocol that the laboratory is using. And of course, we have to remember that there are human beings that are doing these tests. So there can be mistakes because we make mistakes. And so these tests, for the most part, have a specificity and sensitivity of 95%. And one of the examples that I've given before is that if we had like a basketball player that was making 95% of their shots, people would be like coming from all over the world to see this basketball player. It'd be all over the news. So, you know, if there's a scientist in a lab that's making 95% of their shots, like that's pretty good.
Don't even start with baseball. Holy cow. With the mRNA vaccines, do our own cells actually express the spike proteins on their surface? And does our body then destroy our own cells?
Very good question. So what's going to happen is that the spike protein is going to be expressed on our immune cells, and then we're going to make antibodies to that spike protein. And then ultimately, what will happen is if you are infected with SARS-CoV-2, then the antibodies are going to attack the virus, not attack our own cells.
All right, I'm going to take the lull in questioning to ask you a question about the coronaviruses from bats. That was a strikingly high number as far as I was concerned with the number that you showed. Is that, you often hear as bats as a species that's going to be the crossover species, I believe. Is that a number that we should be worried about? And is that one of the reasons there's been such a push from different organizations, including environmental organizations, for preserving wildlife, is to minimize the possibility for that type of crossover?
Yeah, absolutely. And I think that, you know, as we continue to see changes in the climate, as we encroach on the bat's habitat, we're going to continue to see these spillover events or zoonotic transmission. And of course, those viruses are there. We know that they're circulating in bats. They're not pathogenic in bats, but they're obviously pathogenic in us. And so the more that we interact with them and the more that we encroach on their environment, there's always the opportunity for these kinds of events to arise. And so I think that, you know, one thing that we can do to be prepared for them is to be studying them, to be surveilling the viruses that are present in bats, and so that we know and we are prepared, so that if another coronavirus were to emerge from bats, that we would be prepared for it. And it seems like now, because we have the mRNA vaccines, that we would be able to combat it, because we probably could develop a vaccine very quickly that could be utilized. But in the meantime, we probably want to get to a point where we're able to prevent that from happening. And I think that that has a lot to do with human behaviors.
And this is perhaps outside of your bailiwick, but I'm going to ask it anyway. The pangolins are such a weird creature. I mean, wicked cool, but weird. Is this the first time that we're pointing to them as this crossover that you know of? And any ideas why? Spillover, I'm sorry, not crossover, spillover.
Mm-hmm. Yeah. It's the first time that I am aware of that they have been implicated in a crossover or spillover event. And what's thought to have possibly occurred is that pangolins are actually traded in some of these markets. And it's thought that pangolins had been banned from these markets, and so that there weren't any pangolins that were supposed to be in the wet markets in Wuhan, but there is a possibility that sometimes there is some backdoor trading of animals that are banned from those markets. That still does go on. So I think that there's still a lot of question about whether or not that could have been taking place. And I certainly don't have the expertise to speak to that, but that is one possibility. But these animals are treated and they are utilized for various reasons. And so I think that's another thing is, you know, kind of thinking about what it is how we interact with animals in those kinds of areas, like in wet markets and things like that, and how we're using them for human consumption.
And another question, will there be a chance that we would need to start getting the COVID vaccine every year, just like the flu shot? Or if not, will it become less affected over the years without getting a new shot every year?
Mm-hmm. I think that's a great question. I think that lots of experts are thinking that we might need to get a COVID shot each year, just like we do for flu. And the reason why is because this virus is continuing to circulate, the virus is continuing to mutate, just like influenza does. And that's why we get an annual influenza shot, is because the virus is always mutating. And so we try to predict what strain might be circulating, and then we try to use a vaccine to combat that. And it might be a very similar scenario for COVID-19.
So I'm going to do a follow-up with that. What are the odds now that there's been a really significant a show of force isn't the right phrase, a really significant result from the mRNA vaccines that they can be created so quickly and effectively? What are the odds that once we get past this, we'll be able to use that technique for the influenza
I
virus?
think
Do you
that,
know?
yeah, absolutely. I think these mRNA vaccines have revolutionized the way that we think about vaccine technologies. Influenza vaccines are still actually grown in chicken eggs. So it is a very outdated form, but effective form of development for a vaccine. So now that these mRNA vaccines have been shown to be so efficacious and they can be produced on a very large and rapid scale, I think that we will probably rethink how we generate lots of vaccines. And this may also open the door for other diseases for which we haven't had vaccines in the past, so that we can develop some novel vaccines to prevent those diseases. I think, and someone asked a question in Q&A, are most of the mutations that happen in SARS-CoV-2 concerning or significant? And actually, probably most of them that occur are not concerning or significant, because there are mutations that are arising constantly and we don't even hear about them. The only time that they become of concern or something that is significant is if there's a change in how the virus is transmitted, or whether the virus becomes more pathogenic, meaning that it would cause a more serious illness. And so until we see a major change like that, then it would not be considered more concerning or more significant.
Hey, Melissa.
Hey.
I have no idea what happened there. Well, I do, but
It's pretty windy.
nope. Anyway.
Okay,
So
I just answered a question that was in the Q&A about the most of the mutations being concerning or significant.
okay, great. Is there anything else? But I wanted to save a little bit of time. We both have a little bit of a plug that we're going to do for the coming times. Is there anything else with this COVID vaccine? What I forgot to mention when I introduced you is that I had the opportunity we had a forum in a bar for a Science on Tap where you spoke and Maginnis from UNE spoke about viruses and bacteria, and it was November. And I remember when COVID, the whispers of it last January came out, all I could think of was, oh no, I've heard about this. I know it's coming. And it was just, I felt the knot in my stomach get bigger and bigger as the time went on. But I did feel like I really did feel like I knew it was coming. Do you have any sense, just kind of listening to all the information that you listen to, that this might be something that we have to worry about continuously? And I realize the environmental aspects of it are, but does it feel like we've possibly figured out as a whole society how to respond to this better? And what else can we do?
Yeah, so I think we have all figured out how to respond to this better, but we haven't contained the virus yet. The virus continues to spread, and even with all of these measures that we have in place, it's still spreading. And so I think this virus is with us for a long time. I don't think, just like influenza, I think it's going to continue to circulate, and we may have a seasonal COVID shot. We may continue to adapt the way in which we interact with one another. And I think that it probably has shaped this generation, like you said in the beginning. Yeah, I don't think it's going anywhere. Once it's been introduced into the population, I don't think that it's going to fizzle out the way that SARS did or the way that MERS did.
So I guess my little plug and promotion for all that we know about science is face masks are literally all that. And we should be doing that even if you're vaccinated. I happen to know there's a few folks at least who are listening who have been vaccinated. Besides the fact that I'm thrilled for them and slightly jealous, I am also encouraging them to continue to wear their mask, and for us to wear a mask. We still have a long way to go with this. The science has been pretty extraordinary, but we can't do it by science alone. I will coming up in the next sessions, I'll just do a really quick thing before I answer another question, because I don't want to forget this. We have 4 more weeks of sessions that are going to talk about COVID and science responses. So I'm just going to throw that up really, really quickly to, if I can remember how to do this, my apologies. I'll ask the question first and then I'll do it. So, all right, the next question is, do you think that non-medical masks are effective as a medical mask?
So it sort of depends on the construction of the mask and the fabric and basically the fit. So for general protection, you can use something like a medical mask, which I think you're referring to as like a surgical mask, or a 2-layer mask that has a tightly woven fabric. And they are both effective. But one thing that's really important is making sure that you have a proper fit so that you don't have gaps along the sides, making sure that it fits snugly, around your nose, on the sides of your face, and comes under your chin. And so that's really important. And also making sure that we wash our masks, or if you're using masks that are intended for one-time use, that you're disposing of them and utilizing a clean mask on the next go around. And so that's what's really important. There has been some information in the news, especially about these variants, and there could be increased transmission, that people are suggesting that we double mask for added protection. And there has even been some recommendations that have come out from the CDC on that. And I think that it's, what it really is a reminder that we have to make sure that we have the proper type of mask, that we have a good fit, and that maybe when we're in a situation that could be potentially high risk, let's say like you're working with a physical therapist and you have to be really close together, then possibly that may be a time that you might want to wear some added protection.
Excellent. So really quickly, I'm going to share with our audience, these are what's coming up next. Some really great stories from Maine. And then the very last one is a bonus one that we just confirmed last week. So I know there were some questions about the COVID vaccines, but Elizabeth Marnick is going to do a deep dive into all of the COVID vaccines that are available and how they work as our last session. But before that, we'll hear from some companies and organizations. Most of them you've heard of and some you haven't. And I'm pretty excited about being able to share some of these stories. So if you go to the mainsciencefestival.org website, click on the very large COVID molecule picture, and that'll take you to the webpage. It has this information where you can also sign up. It's also online on all the social media spots. And Melissa also would like to do a quick plug, a very important plug as far as I'm concerned, for what they're doing.
Okay, thank you very much.
Yep.
So the University of Maine is our state's research university. It is a land, sea, and space grant institution, and it's the flagship campus of the UMaine system. And UMaine offers over 100 degree programs that are combined from undergraduate up to PhD. And our graduate students and undergraduates are engaged in awesome research learning experiences that help to prepare them for the workforce, and also helps to train our students to become innovators and leaders in their field. And so the University of Maine really weaves our students right into the fabric of our university, and the students are working with our world-renowned faculty to generate new knowledge and develop solutions for problems that impact people in Maine and beyond. And so I'd like to invite you to take part in the UMaine Student Symposium that will be taking place this year virtually on April 16th. And this is actually part of Maine Impact Week, which provides an opportunity for the public to see all of the efforts going on at Maine's Research University and how we are contributing to the economic advancement of Maine and beyond. And the UMaine Student Symposium really is the culmination of our Impact Week. I invite you to look into Impact Week and to attend our symposium, because our symposium actually has a COVID-19 theme this year. So it's the role of our research university in response to crises, with really a focus on COVID-19. And this is the agenda for the student symposium. So we're going to have a panel from Maine business leaders that have been involved in the COVID response, very similar to what's going on with the Science Festival. Those from Abbott Laboratories, the Jackson Laboratory, and Puritan Medical Products. We'll have student presentations in the form of flash posters. And then our keynote speaker is Dr. Nirav Shah. the director of the Maine CDC. And then there will be a students award ceremony. And actually for 2 weeks leading up to the student symposium, you'll be able to view the students' presentations, their posters and oral presentations on the UMaine Student Symposium website. So I invite you to take part in this wonderful event. We really would love to see more participation from the public so that you can see the excellent work that's going on at the University of Maine that's really driven by our students. Thank you.
All right, and that is a wrap for this week. We're going to have another session next week at 1 o'clock where we're going to hear from Abbott, Jackson Laboratory, and IDEX. IDEX's story of how they've responded to the pandemic is different than others in some ways, but no less exciting. And to follow up just quickly on what Melissa said, the University of Maine is a hidden gem in this state. She didn't ask me to say this. I'm just telling you as one of the inspirations for why I started the festival. They are doing extraordinary work on many levels, work that will stand anywhere. And I think one of the most remarkable things that you will get if you pay attention to what's happening at UMaine is that the undergraduate students are deeply involved in the research in ways that matter. It's not just washing dishes. So the opportunity to hear from students, both undergraduate and graduate, if you have the opportunity, I recommend it highly. And that is it. Thank you very much for joining us. Thanks again to Bioscience Association of Maine and the Jackson Laboratory for sponsoring this, and Maine Public. And hopefully we will see you all again next week. Thank you.
Thanks, everyone.
Thanks, Melissa.
Bye.